Can you guys see my screen?
Yes,
Yes.
you can put it full screen.
Yeah?
Great.
Is it good now?
It's good.
Okay, thank you guys for waiting. I was having some technical difficulties. Okay. Good morning or good afternoon, everybody. My name is Chioma Schurman. I am a student at the Anton University of Suriname studying biology. And today I will be telling you guys about the inherent variety and acoustic monitoring potential in Suriname. The research presented today was done by myself and Note Ten Half, who couldn't be here today, under supervision of Professor Slabakorn and Dimitri Oboter. Tropical ecosystems support some of the highest levels of biodiversity on Earth and are exceptionally important for the conservation of global diversity. Tropical rainforests make up about 7% of global land surface, and of that 7%, 90% consist of rainforest species. These systems not only regulate the global climate and act as major carbon sinks, but they also harbor about roughly half of the world's plant and animal species. Within our tropical rainforests, we have very high species diversity compared to other parts of the world, of which amphibians represent the most diverse vertebrate groups. They play an essential part in ecosystem functioning through their central role in food webs and nutrient cycling between aquatic and terrestrial systems. Amphibians are, besides that, important um, because they are good bioindicators due to their sensitivity to disruptive environmental changes. So they can react to the slightest changes in temperature or humidity, making changes as they happen relatively easy to spot in these communities. Population declines, therefore, can signal action potential before other species may be subjected to that. They are also especially important for us humans because with amphibians, their diet consisting of mosquitoes and insects, they can keep those populations relatively stable, in which they lower the chances of, for mosquitoes, transmission of diseases like dengue and malaria. However, due to a bunch of issues, the global population has been declining since the 1950s, and it has not yet stabilized. Global threats that these guys are facing include habitat changes like deforestation, pesticides and environmental toxins, from, for example, the gold mining industries, alien invasive species that can cause resource competitions with the native species, emerging infectious diseases like chytridiomycosis, and overexploitation that can happen because of the pet trade, for example. Monitoring is the systematic process of collecting and analyzing data and using that data to track within the field of biology, for example, environmental changes or observing species in their natural habitat and their behaviors. Different species differ in activity, location, like in trees or on the ground, for example, which could make it a challenge trying to map communities as a whole. By doing physical monitoring, there is also a certain level of disturbance that we subject the animals to just by being there. So our presence is a disturbance. The tropical rainforests can be quite inaccessible due to their density, which is also a challenge. The temporal scope is long-term monitoring, and for that, physical monitoring would cost a lot of time, energy, and of course, money. Which is why ecoacoustics or passive acoustic monitoring in this case for amphibians, for frogs, could be the solution to all of these obstacles. Passive acoustic monitoring is a non-invasive approach because the recorder is placed and then is left for longer periods of time. It is quite feasible in the long term because this equipment uses batteries and the monitoring can therefore be continuous. Not all species vocalize, but research shows that vocal diversity gives a rather reliable estimate of the total biodiversity, which is why this should be utilized in monitoring. And we have chosen frogs, anurans, because they have a high diversity and because most of the species vocalize, which gives us a higher chance of identifying them based off of just sound. Monitoring in Suriname. In Suriname, there are a few organizations and institutions that are involved in monitoring projects and the execution of these, which include the National Zoological Collection of Suriname, the Anton University, and Leiden University, and especially the BioREPS program, which we are a part of, and Neo Wild. In the Amphibians of Suriname, published in 2012 by Albote and Jairam, we can see the parts of Suriname that have been explored and where all of these species reside. But as you can see on this map, there are a few areas that have not been covered by these red dots, which indicate the amphibian sampling locations. This does not mean, of course, that there are no frogs there. There just hasn't been any research done in those areas, which is why monitoring is important. And this graph shows us that there is a constant growth of frog species described in Suriname, which is why monitoring is important because there is much more to be discovered and described. Invasive species like the Eleutherodactylus johnstoni and Eleutherodactylus planirostrus have been these guys have entered the Caribbean in the last 30 to 40 years. There's a sound. Oh, wait. I'm trying to play the sound, but oh, here it is. Wait. Can you guys hear the sound?
Nope.
No.
No. Well.
Did you share it at the start, the sound, or not?
Yeah, I think so. Let me check. You guys are probably not going to be able to hear it, but I have my phone here, so I'm gonna play it manually. One second. Can you guys hear that?
No.
No.
No? Oh, okay. Well, Okay, well, we're just gonna continue with the presentation. Yes. So this is a map of the Caribbean and where these invasive species have spread in the last 30 to 40 years, but not only in the tropics, because in Europe, some of these invasives have already formed stable populations. In Brazil, for example, there has been research done in which shows that these in which it shows that these invasive species are competing for resources with the native species and that they are driving them out of the areas where they're endemic to. There are no published articles about the presence of these invasive species in Suriname, but together with one of our supervisors, we have observed both of these guys ourselves in numerous locations. This lack of research in Suriname leaves us with limited knowledge on the impact of these invasive species on our native anurans. The research objectives for us were first to assess how accurately ecoacoustic data can be used to identify anuran species and monitor communities. And this was the monitoring part of the project that we did. And our second research objective was to evaluate how acoustic behavior of invasive anuran species affects the calling behavior of our native anurans. And now we're going to dive into the first research objective, assessing how accurately ecoacoustic data can be used. As for our equipment, we used audio mods, and this was standardized by hanging it up at approximately 1 meter 50, which is around breast height. For the data analysis, we used Audacity and Raven Pro for the visualization of the audio and for the identification of species. And we also used Seno Canto as a reference for certain frog sounds. We monitored various locations, including the Fredberg area, the Fredberg Base Camp, and Fredberg Mountain. The duration of this monitoring, the data we have gathered was around 48 hours, sorry. The habitat of Fredberg area is high dryland forest, and the season was the rainy season. And I should mention that the rainy season um, is especially valuable for us as researchers to do research on amphibians because that is when most of them reproduce and they are also vocally active during their reproductive season. Our results.
We can't hear it.
You can't. Oh, man, that sucks. Hold on.
You can maybe try to stop sharing your screen and first of all, maybe download the presentation.
Yeah, it is.
Because online is always with issues. And then before you share your screen, see to the left where you see on the right of share content, you need to include sound.
Yes, I didn't have that
So
activated.
now if you share it, hopefully it'll work.
Okay. Okay.
Yes, let's try it again. Yes, we can hear it.
Great. this was what one of the very beautiful nights sounded like at Fradberg. And if you are not familiar, these are all frog sounds, mostly that you're hearing. And they will now be presented in a table. Yes. These are among some of the results of the species that we have been able to identify at the Fredberg area. However, the data analysis is not yet complete because it involves a lot of hours, a lot of listening, and very intensive. But here we have a snippet of the Boana calcarata. And next we have a snippet of the Rebogutatus. And you can also see what the spectrogram looks like when these guys are calling. We had two more locations, which were Palulu Camping and Sandre Ain. The habitat for these two locations is savanna, and the duration is ongoing. Within the 2 months that we had for the research, we went back previous times to Palulu camping and Sandere to employ audio mods. We went back to pick them up, collected the data, and before the 2 months were over, we went back one last time, employed the audio mods again on numerous locations within the area, um, trying to pinpoint specific communities or other things that we had in mind at the moment. And that is still ongoing. These audio mods have yet to be picked up, and the results of these areas I will not be sharing today because of the limited time. So for our playback experiment, we focused on the native frog, Leptodactylus fuscus, and this is what he sounds like. If you've been in Suriname at night, I'm pretty sure you've heard this guy. What we did for this experiment, oh, wait. Now I don't know how to stop it.
It's fine.
Okay,
We
but
can
you
hear
guys can
you
hear
through
me
it all.
fine, right?
It's fine.
Okay. Okay, great. So we did a trial for this playback. We had 3 types of stimulus. The first stimulus was a call of the Johnstoney, which I unfortunately could not let you guys hear at the time. But the Johnstoney is the invasive frog that we wanted to test the impact that this frog has or could have on our native species. As a playback stimulus number 2, we had a control of the Leptodactylus fuscus. And as stimulus playback 3, we had a sound of the Rina La Marina, or within the soundscape of the Rina La Marina, which, should you know, is below the frequency that the Leptodactylus fuscus and the Leptodactylus johnstonii both call within. So for this trial, we did a pre-trial silence. So what would happen is we would identify calling individuals. We would put our microphone at a standardized length of 5 meters to record. We had our JBL speaker, which would play the stimulus at a standardized length of 2 meters. And so what we would do is that we would find a calling male. We would get close enough for our recording, and then we would do 3 minutes of pre-trial silence, which would be our baseline for what the calling activity is like. Then we had our first stimulus playback for 1 minute. Then we had a silent interval of 3 minutes. Our second playback for 1 minute again, 3 minutes of silence. Our third playback, and then another 3 minutes of post-trial silence. And that silence would just be us recording again for 3 minutes, trying to see if there are any other changes happening. The location was the Anton University campus, and this was chosen due to its accessibility to us as students. These guys are all over Paramaribo, but when we were scouting for this research, we would be driving by and it would be on people their yards, which we don't really have easy access to. The duration was 3 weeks, and the analyses were done with Python and Audacity. And here you have some pictures of us in the field. So the results of the playback here on the left, I have included the 3 frogs that I just mentioned. And in this graph, you can see the individual shifts in calling activity. This graph shows the response to Johnstone and Fuscus control playbacks in ratio to the baseline. Statistical tests like Wilcoxon and ANOVA were done parametrically and non-parametrically to give us our significant values, but this shows that they do not have significance. There is, however, an outlier, which is 3001, which is the pink one. And when this gets included into our analysis, he goes against the trend of the rest of these frogs, and which skews the graph. When he gets ranked, the rank weighs a lot due to our small sample size of 7. This graph shows the shifts in dominant frequency, and it shows us that the dominant frequency stays constant among individuals. The same statistics as the previous graph were done for this. And there are, again, no significant differences because the frequencies of all the individuals are relatively the same. So for our discussion, ecoacoustic monitoring should be done respective of the research objectives. When you are trying to monitor communities, you should try finding areas that harbor these communities and that can kind of show you the interactions that they have with each other. And passive acoustic monitoring is great for that because you can have continuous monitoring and you can really see everything as it happens, and also in the hours of the night. For our playback experiment, we should have had a bigger sample size. Like I had already mentioned, this was a bit of a challenge due to the location. We only really had full access to the university campus, and we also had, I would say, limited time. Again, there was an outlier which could not be which there was an outlier, and for the statistics, we would just need a bigger sample size. There have been differences in the calling behavior of specific individuals, which doesn't mean that we can't find a trend in later research. You also have the concept of bold versus shy individuals. Certain individuals reacted way more quote unquote aggressively than other individuals. And for the dominant frequency, you have morphological constraints because frogs only have a limited range for their dominant frequency, which is affected by size of the individual, for example, and so it is physically impossible for them to call louder or have a louder dominant frequency than they are just genetically bound to.
Chioma, quick reminder.
Yes.
We have 5 more minutes before we move on.
Okay. So what we have concluded out of this presentation is that there should be continued monitoring because we have seen beautiful results for the long-term monitoring, and we have been, and it has been effective in IDing certain species. For playback studies, we need to have a bigger sample size. Of course, we have limitations when it comes down to the species that we are doing the research on. And of course, there should be future research done. Do you guys have any questions?
Thank you. Please raise your hand if you have questions or just ask. That's also possible.
So maybe I can ask a question about the playbacks, Chioma, because what you showed was that the species that you tested was responding to its own call by lowering its activity as if it's listening for a particular competitor. And the other two species, one invasive, or were they both invasive? No, there was one invasive and
No.
one. They were both invasive?
No. Only one. The Johnstoni is invasive.
Yeah, and the Marina is low frequency and not overlapping, but both of
Yes.
them were actually also having a similar effect than playing back their own species. So does that yield at least the suggestion that there may be an impact in terms of competition?
Yes, because like I had said, there have been individual differences in the behavior of the response in the individuals that we have tested. And so yes, this could mean that they have a certain similar reaction to the invasive frog and have less of a reaction to the sound they know, the control, and the other frog, the toad, which lies beneath their calling frequency. So we would in for future research, it would be ideal to have a bigger sample size so that we could see this possible trend a lot better.
Yeah. Yeah, because I was thinking that these calls in most of these species, I do not know much about the frogs, but they aggregate based on calling, and so many males approach calling aggregations, and then they will attract females by the compound aggregation of calls. But then, of course, the second stage is that they need to stand out, and females need to choose them instead of the competing males. So when I was wondering, well, how the invasive species and the calling in such in those two stages will have an impact, because now you tested actually the impact when the aggregation has been completed, so that it joins an aggregation of already present males, I guess.
Yes, and there's a lot of spectral overlap because these guys call within the same frequency. So yes, definitely very interesting for future research.
Yeah, because I think females, they can be attracted first to the aggregation and then they make their choice, but it could be that they actually listen to any call that indicates a suitable habitat. And so then the invasive species may also stand out. Okay, this is good anyway. There's individuals of my own species and other species, but I think that's for future study, I guess.
Yes, it is.
All right. So thank you, Chioma.
Yeah.